In the first video, we saw that we can find an insulin gene in each person, and that the insulin gene is nearly identical. Um, and so the BLAST search algorithm finds it as the best match by far in the entire genome, but there are slight differences between the genes. The differences between a gene that are found in different individuals in a population are called alleles. And so in a population of any species, we have a variety of alleles represented. There are natural alleles that occur in a population. Those natural alleles are what give rise to all the heritable differences between us and any species. And developmental biologists take advantage of that and compare the alleles of developmentally important genes in a population to learn more about what the job of that gene is. Random mutations in nature give rise to different alleles, and those random mutations are called spontaneous mutations. In addition to the naturally occurring alleles, developmental biologists create new alleles in lab by inducing mutations. So they'll expose fruit flies to, say, X-rays and increase the rate of random mutations in order to produce new alleles. So these are not naturally occurring alleles, and developmental biologists use the creation of new alleles and the analysis of those alleles as a way to understand what the function of a gene is. So for example, imagine in our insulin example that you didn't know what insulin did. And so you studied it in people, and you saw, oh, there's some people that have defects in the insulin protein and are diabetic. So we're learning something about what insulin does. In order to understand that better, imagine that we could take people and make all kinds of different insulin alleles to learn in greater detail what it does. How does it do that job in detail? And developmental biologists do that a lot and produce lots of alleles, basically lots of versions of the gene that are defective to different degrees. There's one special allele that we'll talk about in this course a lot called a null allele, where the function of the gene is completely destroyed. And so that's a null allele, and that's special because they got no insulin. So none of the insulin function is present in that person. We can really use that to help understand what are all the jobs that insulin does in a person. When scientists do this induced mutation in this genetic analysis to produce new alleles, they have two different ways to analyze it. One is this first type is the traditional genetic analysis. That could be done for a spontaneous mutation that occurs in nature, that maybe somebody brings in a lamb that just has one eye, and scientists could study that lamb and learn about what gene was mutated in that lamb to give it such a bizarre phenotype. They could also study induced mutations in a lab to find out what mutation gave rise to such mutant phenotypes. Developmental biologists often do something called reverse genetics, where instead of starting with a mutant phenotype and finding the defective gene, they start out with a gene, they damage the gene, like in a test tube, they modify the DNA sequence, put it back into an organism, and then find out when that gene's defective, what does the mutant phenotype look like. When doing reverse genetics and producing these modified organisms that have intentionally damaged genes, those animals are sometimes called knockout or knockdown animals or plants. The in putting a gene back into an organism to study it, we produce a transgenic organism. When a null mutation allele is put into a transgenic plant or animal, that's sometimes called a knockout mutation. We'll talk a lot about mice as examples in this course, and mouse geneticists talk about knockout mice frequently. There's a newer technique we'll talk about a lot in this course where instead of mutating the gene, scientists are able to block its expression using a naturally occurring mechanism called RNA interference, or RNAi. And so instead of knocking out the gene, they say they knock down the gene. And again, that's kind of a casual term, but it's used frequently by developmental biologists. They'll knock out a gene and study its function, or they'll knock down a gene and block its expression. In your assigned reading, there's a box that describes RNAi in the book, and the figure shows the natural gene regulatory mechanism that is used to repress expression of target genes. And so developmental biologists take advantage of that naturally occurring pathway, and they will introduce new double-stranded RNA, new blue molecule that will match to a target molecule and target it for degradation. The red mRNA, for example, could be from the insulin gene, and we could knock down expression of the insulin gene by introducing a double-stranded RNA that was complementary to the insulin gene, and that would target the insulin gene mRNA for destruction. In a knockdown experiment, the gene is wild type, or normal. So we have a normal insulin gene coding a normal mRNA, but the protein doesn't get made because before the mRNA exits the nucleus, it's degraded. And so the knockdown is functionally comparable to the knockout. In one case, we knock out the gene so it can't make a functional protein. In another case, we knock down the gene expression so it can't make a functional protein.